Omkar sir, you have worked as a COVID warrior during the COVID-19 period. So I would like to know your experience of as a RT-PCR technician and in a more detailed view of the process that you carried out from sample collection to the result interpretation.
So, to start, thank you very much for having me as a speaker on this panel. So, to be precise, let's first of all let me express my linkage to this particular position as a RT-PCR technician in this COVID-19 response target or response force, what we could call. So, basically, I have done my master's in applied genetics and real-time polymerase chain reaction. That is the acronym we call it is the RT-PCR. Or the other suggested acronym is like explanation to this acronym is reverse transcriptase polymerase chain reaction. Uh, this is a testing systematics we usually use for different like viruses or any genetic code, specific targeted code to identify these codes. So as a master's in applied genetics, this been the part of our practical as well as the part of our academia. So that is how I just like in the initial phase I was very familiar with the application part of this test even before the common people would get knowledge about this particular test. Coming back to the particular RT-PCR test and its implication in the uh, COVID-19 particularly, if I have to consider. So first of all, COVID itself means coronavirus. We can in general term, and as it this pandemic appeared in 2019, so we call COVID-19 pandemic. And the pandemic specifically means the spread of any disease across the continents. That is we call pandemic. Now, as per my role, so basically, as we have given as a part of diagnostic labs, what one rule you have to like basic rule you have to follow is to follow the tat is turnaround time. So that stands for as soon as you receive the any sample in your labs, in how much time span you are going to deliver the results to the particular uh, patient or the expecting personality on the other side. So in this setup, what was proposed by the research facilities or the faculties or the authorities we are having at that time. So the expected TAT was turning around time is 5 to 6 hours. It was including collection of the sample, transportation of sample to the diagnostic labs, and then further delivering the reports or generating the final reports. So in case of uh, setup what we having in our city particularly, we were divided this whole setups in three facilities. So first facility was sample collection facility, second facility was diagnostic lab particularly, and the third facility is quarantine facility. Okay, so what we are doing, if at all one patient approaches to the like centers where we have established different preliminary healthcare centers we were having, and there we collecting the samples from the particular patient. So patients comes up whether it is required for the particular symptoms he showing, or whether he's traveling somewhere, so he require the permission for his travel. So there were different reasons for which you were receiving the patient which having like mixed population. They might be symptomatic or there might be asymptomatic. So in the both cases they come to us and we have to take the sample. Now what is sample taking procedure here? So as it is COVID-19 like coronavirus, so it is expected to have infection or it infects to particularly respiratory tract or respiratory system of the host, in this case like we humans. So here what happens when you get a infection of COVID-19 virus, you show a similar symptoms of that of influenza, like coughing, blocking of your cough in your particularly chest area or the lungs area, and not particularly choking of nasal cavity and the mucus in nasal cavity, but particularly having the cough saturated in your bronchitis, like bronchial area. And what body respond in the action is it usually try to throw it out just to clear the tract, in order to breathe well. So in that case, you basically that whole procedure we call cuffing in the basic sense. So during the cuffing, the viral particle, if at all it has virus particularly, so those there are high chances that it will get stuck to the mucal layer of your pharyngeal cavity. Now, what is pharyngeal cavity? Where our oral cavity ends and connects with your nasal cavity as well. So that entire the session of our end of our oral cavity, just before your bronchitis or bronchial opening, there is pharynx, and above the pharynx we have pharyngeal cavity. So the sample for the better like having better diagnosis, you take two types of like sampling, and sampling is done by the swab. Now, what is swab? So swab is a plastic like stick is there, and to the sticks we have synthetic threads at the bud of synthetic threads at one end, and this has particular length of your oral cavity in general, so around 8 centimeter, 8 or 10 centimeter approximately, and it usually touches to your pharynx at the end. And so you have two methods to take the your sample. One is nasal pharyngeal swabbing, and another is oral pharyngeal swabbing. So one swab you will take the sample from your pharynx through your oral cavity, and one swab you will take sampling from your nasal cavity. So that the two methods, or sometimes you can have both of them.
In some of the countries they took samples from like anal samples, right? So can you explain me why they particularly taken samples from anal route?
Annual route, I guess, like I'm not the like I am not familiar with this procedure particularly, but if at all I have to justify it like clinical point of view, so I guess they are targeting the mucal layer of your openings. So possible openings where the mucal layer, why mucal layer, like mucal layer of different openings. So why body like produces mucal layer in first place is to keep the moisture over there, whatever the place, like entrance you have nasal cavity, you have oral cavity. So first thing is moisture to the skin, that particular area. Second thing is it also act as a barrier for possible infections. Okay, so I guess when virus got infected in your like lung systems, and lung is the like shortest route to deliver the viral load into your blood. Okay, so now through the blood the viral particles are traveling throughout your body, and whichever the area where the possibility of exchange of your blood system is going on. Now in the anal cavity or intestine cavity, we could see that the blood is actually circulating for the exchange of nutrients, or in the rectal cavity the exchange of or control of water particularly took place. So in that case, in my opinion, the virus get, if at all it is in substantial quantity in your body, in your blood, so at that time it just get attached with your mucal layer, and through mucal it body is trying to push out the infection like it is doing through your lungs way, right? So in that case I guess they are trying to catch that.
But
But
I
depend
but
actually
I think having a nasal and oral route which is successful for that process,
yes, particularly
I
let's
don't think we
focus.
need to don't
Do
stretch the
yeah,
topics in that
but
much.
I guess there are like apart from like RT-PCR, you are actually taking mucal samples from your nasal and pharyngeal cavity, right? There are some other where you can use blood serum, and you can also go for antigen and antibody test sort of, but the sensitivity is very less, so usually it will not be preferred. In the spread when we are having in the like tremendous way around, like it is becoming the pandemic, so in that case the sensitivity matters the most, because the virus, the spread of the virus, probable route, yeah, most probable route we could say. And also the another thing is, along with the your sensitivity, it is also giving you very short turnaround time, so you are giving accurate reports in the short terms. And it also very sensitive, like even one or two viral particles are there, if it detects, it can trace it out. So that was the idea behind preferring the RT-PCR through your mucal or nasopharyngeal or mucal, because usually in when you get into the people and like around the people who might have the infection, and it got the ideal route for coronavirus to enter in your body is usually through your breathing system. So that is why the ideal or the more possibility thing behind it is to have the swab from your uh, nasopharyngeal cavities. Yes. So after that, I will come to the point where we have collected the swabs, right? So now these swabs are particularly have to maintain into the viral transporting medium, because you have viral particles and those particles are very fragile, and you have to maintain its integrity until it reaches to your diagnostic centers, right? So for that you are having VTM, that is viral transporting medium. Now the container of the VTM is like in our strategy, the authorities have chosen, like very carefully they have chosen the tubes of centrifuge. So centrifugal tubes has been used to carry the viral transport medium. Now the idea behind it is basically very simple. When you are taking the nasopharyngeal swabs, so at that time the possibilities are much more that a person is having like other cold or other infection. So in that case he might get some other infections or other uh, contaminants, you can like mucus and other stuffs like that. So as soon as you drop reaches to the sample reaches to the diagnostic center, the first thing they do is do the centrifugation, so such particles will get settled as a pellet at the base.
What is the composition of VTM?
Yeah. So coming to this VTM, the VTM basically is a saline type of saline, and the saline is combined with your Bowen serum albumin to contain the integrity of the viral particles. Then we have pH indicator. Then in some cases we also have a few basic antibiotics. Now these are the contents of your VTM.
Why there is need of pH indicator?
Yeah. So pH indicator, in case you get any other, just the best content is saline. Okay. So in the saline, if you get any other contaminants, whether it is fungal contamination or you get bacterial contamination, so both of them will consume the sugars from your saline, and the sugar will get converted into the acids, and that
Sugars are there in VTM?
uh,
Sugars are there in VTM?
being the part of saline preparations you are having. So yeah, in some case like basic saline we are having, so that saline they have used, so there will be the chances of having some glucose particle, if at all it's not there. So the part
Because you mentioned about Bowen serum albumin.
yeah, the role of bovine serum albumin is as it is serum albumin. So bovine is like basically cow or cattle's and their serum, and albumin from their serum we are using it here is uh, like in virology, just sir explained us, if you have to do like a content living virus or viral particle is intact, so in that case you require to provide it like in vivo condition, right? So Bowen serum will provide it as a sort of, not exactly but sort of in vivo conditions where it will maintain its integrity of stability to its
Stability of what? Do virus require saline solution to be stable?
no. Saline solution, like saline is used for like fluidity is of the medium. Why you require fluidity? Because you're going to dip the swabs into this medium, right? So swabs are basically, as I explained, it having a bud, cotton bud. So cotton shows porosity is there. Viruses like mucus is got stuck to this your bud. Now how you are going to take out that mucus without touching it? So in that case you require some liquid where you can shake it, or you just mix it properly. So the when you shake and mix it properly, it will like get dissolved or flown out of that medium to the your cell.
Does it have to deal
Say
with
Bowen serum albumin has been provided in the VTM. Okay. So my question is that whether the RNA extraction it is completely from the viral particles, or the cells which have been infected with the coronavirus, they are also source material for the RNA virus or coronavirus genomic content?
They can be, but it is
See, when you are collecting sample from the nasopharyngeal region, it is quite sensitive region. So along with mucosal layers, some cells are also going to be part of it.
the cells when you are picking up it, like it will be depend upon, if at all the, I believe that if at all there are some like rashes or something, some wounds are there. If at all you are like, if when you are scooping the sample, like you are taking swab, the swab is not literally scooping. Have you seen the swab? So you would have understood. So scooping is not going on. What you have is like spongy material. Okay, so what it has to do, it has to take the adhered solution. You are not scooping. How much cell line or cells like you are going to collect out of it? That's the question. And
See,
in addition to that,
when you
no,
specifically talk about mouth,
yeah, mouth.
generally the life cycle, it is continuously in the growth period, okay?
Okay,
So cells are continually dividing,
yes,
and this is the region where stem cells are more involved
okay,
as compared to all other regions. So taking the sample from a sensitive area, there are chances that along with water particles, cells also be going to be part of it. Okay, so VTM consists of albumin. Okay, so I don't know whether how that RNA extraction process is going to occur, but my question is that whether you are targeting only the viral particles or also the infected cells.
like eventually if at all your target is obtaining viral genome for the RT-PCR diagnosis, that doesn't matter whether it is coming from, is that really matter in this case? I don't think so, that it is coming out of direct viral particle or it is going to come out of a cell. That's my like point of view to see this situation. My point, like I am trying to explain, is that the role of like,
The target in this case is only viral particles.
no, I don't said that. The target is to collect the viral particle, whether it comes as a viral particle directly, which has been trapped in your mucal layers, which
or
came
along with the cell.
yeah, along with the cell.
Okay, so bovine serum albumin has been provided for the animal cells to live into that.
Yeah,
And pH indicator is for that purpose. If bacterial infection occurs to plant, if to animal cells, the pH will shift and you will visually kind
and another source of like just to mention, the another source is whether when you are picking it up, the definitely you are having your mucal layer, mucus itself, like sugars, no, after all, am I right? Mucus itself a sugar, yeah, the all is chains of mucal itself a mucal layer.
Basically,
They
it
are
contains lipids, right? Lipids.
no
Mm,
sugar.
Sugars.
But yes, as he mentioned, that there will be the cells along with the infection.
I'm not denying. My point of view is,
No, my question is that bovine serum albumin, which has been used in the VTM, does it has a relation with animal cells? That they should also be in a state where
yes, yes,
that genomic content also is targeted in this case, because again it is a sensitive test, no?
definitely, no, it might be definitely 100%. I am 100% like convinced with that. Okay, my point, like my point is basic that uh, we have to see, we have, you cannot like I am targeting the cells particularly, so I have to compose the VTM solution like this. Okay, some company might go for like we want without the lesser cells you provide, the lesser chances of contamination. Are you agree with that? Right. So in that case, why go to that strategy only?
Now let's move
My
on.
point is,
Let's
yeah,
move on.
no, it is because of why. My point is, our target here is obtaining maximum numbers of the viral particles. So whether it is come from the infected cell, whether it is come out of your mucal cell, or whether it is come like from the blood itself, like it, I have a rashes or something, and it's like come along. I am considering all the possibility, and accordingly, I am designing my strategy for your viral transporting medium. So
Okay, now
because of that only, now viral particle doesn't need your antibiotics. Antibiotics is basically with the possibility we have added that there are possibility of contaminations. That might be,
company has added.
yeah, there might be this contamination, may be patient originated. That contamination may be the one who is taking from his uh, handling style. So that might create the contamination. The contamination can also be possibility at the diagnostic center as well. So considering all those things, they have composed this particular viral transporting
Okay, so
medium.
further there is centrifugation.
Yeah. So no, not centrifugation. At the I just come to the VTM. So when you start to collect the samples, the first precaution you have to take is you should wear a PP completely, and the other who have background must take the care of uh, having their exposure to the viruses or infected patients, like that, like having a N95 mask, continuously stir your hands, and like those things protocol has to be followed. Once you got your VTM, you like packed serum and your viral transporting medium, and it is into your centrifugal tubes. Those tubes has to be sealed into biohazardous packaging, and those like with that packaging you have to transport into a chain box. And that chain box, the temperature of the internal temperature of this chain box required to be maintained under 2 to 8 degrees Celsius, just to like avoid the disintegration of viral particles, particularly due to the temperature. Okay, so once we prepared it, then it will be transported to the diagnostic center. There they will sort, they will check, the first team will check it. So let me brief out the internal like partitions of or the parts of the diagnostic facility. So you have receiving facility, then we have nucleic acid treatment facility, then we have RT-PCR facility, where we generate the like analysis nucleic acid and generate the reports. So mainly these three parts are considered in the facility of diagnosis particularly. Now at the receiving center, we receive the your VTMs, and VTMs will be first of all observed for any leakage or any contamination or anything happen to it, and accordingly it will be processed further. Like as I said in the start itself, we will centrifuge, remove the any dirt or something is there, it will get settled down to your pellet level, and the VTM will be further stored at their level. Then further those who have been stored will be labeled accordingly. Somewhere they use a numbering system, so the identity of the patient will not be exposed. So for that, numbering systems, or some have barcode systems, depending upon the like systematics what they are going to follow. After there, it will come to the processing into your nucleic acid extraction center. Now at the nucleic acid extraction center, what the first thing we do is we divide this VTM particularly into two parts. So one is kept as a backup, and another will be further taken for the processing to extract the nucleic acid out of your uh, transport medium VTM. So there we have, like, first of all I have to tell you the material, like basic material required for your nucleic acid extraction. So it is like, well, first thing is lysis buffer, second is magnetic bits, third is wash buffer, and fourth is elute buffer. So these four material basically is there for the material right now I explained you is a part of manual method of nucleic acid extraction part. So there are like semi automated system are there, there fully automated system are there. The only difference is they lower the labor work and the hustle you require while doing these treatments. So also they help you to treat multiple samples in the at the same time, so that saves you multiple time, like time which is required for your nucleic acid extractions. Okay.
Can you
In
explain about lysis buffer
Yes,
and
yes, I will come into that
okay.
right away. So the chemical, let's talk about the compositions particularly. So in the lysis buffer, as it already named, lysis buffer is particularly like aimed to lyse the viral particle, target to the lysis of your viral particle particularly, and the lysis of that particle will allow the nucleic acid, whatever it is having, that relevant nucleic acid to get outside of its particle viral capsids or composition, whatever it has. So out of that, it will flow out of these in your medium what we are right now having there, and that medium will contain your this viral material, and that viral material furthermore will bind to your magnetic bit, and magnetic bit will contain for a while. Okay, and whatever the rest of debris over there, cell debris, cellular debris you take, or viral capsid debris are there, which will be washed off by treatment of your wash buffer.
So
Coming to the
Sorry, sir. To
I
interrupt
will
you.
I will just finish his question first of all. So there are certain chemicals like guanidinium thiocyanate sort of chemicals, which usually act as a lysis, particularly work in the lysis of particles, viral particles.
It goes and bind, right?
Yeah, yeah, and they will like break down and break open it. Yeah, now coming to your point that yeah,
Magnetic beads particularly interact with the genetic material of the virus.
yeah, genetic material of
So,
virus.
is there is any binder or any kind of complementary sequence which exactly bind to the viral DNA?
As per my knowledge, there is no direct sequence binding is going on. I think it is basic chemistry of like variation in your PhD and surface
Maybe
adherence. Okay, so it has the magnetic bit, particularly composition of one magnetic like magnets, like magnetic particles, and that particle is coated with like silicon like material. Okay, so the surface of that silicon like material shows affinity in
towards the
towards the nucleic acid
Hmm.
in different medium, differently in different medium.
Okay.
So if you have like, for example, it basically forms, and if I have to like go in very deep, so it basically forms hydrogen bonds with the surface in the influence of when you have a number of like acidic medium. So you have already slightly more acidic, is not in the sense of like buffer acidic, lightly number of hydrogen atoms are more in the medium, so it will like okay bond, create and a temporary bond with your surfaces.
H plus ions basically they
Yeah.
together.
By changing the
What is the exact interaction between the DNA and magnetic bit?
Yeah, can you just
Yeah,
explain details?
if I have to simplify it, so basically as we know, it also have sugar backbone. Any like you take RNA or you take DNA, so it has a sugar backbone. So as sugars we already know, it is very soluble in the water, right? So how the sugar gets soluble in the water? When we say it chemically, so what exactly chemically happening? So water molecules actually by creating temporary hydrogen bond, and hydrogen bond is the weakest bond.
we can say that there is a phosphodiester bond also is there. Phosphodiester bond.
Hydrogen is not the weakest bond.
Van der Waals.
Van der Waals, yeah.
Even water wall is not the weakest.
So it is a
It is a cumulative bond. If you're talking about water,
Not the weakest, I mean weaker bond compared to that of other ionic bond.
it depends actually. If there is cumulative force, it is the strongest bond.
Yeah.
Okay,
When they come together, then it's strongest one.
so in this case I like I understood like this, that how you like sugar gets soluble in your like water, how it got so basically that solubility can be influenced by the influencing medium, right? So sugar is solute and water is solvent, and influencing the content of your solvent, you can influence the solubility of solute. That is the basic principle. So in my opinion, I guess binding of your nucleic acids are usually work on this principle. Why? Because that makes it easy for the elution composition of your elution buffer. The more simplest you keep it, the more easy it will be for your composition of your elution buffer. So I think like while designing this side of like, it's not my research to like prepare these all chemicals. Usually it comes as a kit. So the basic principle and idea behind it, how they are understanding, so accordingly I have understood to it, and that is how I am explaining. So in my opinion, silicon coating is there, so it got wrapped by simple this kind of forces. So once it got coiled around your magnetic bits, then you get wash buffer treatment. That wash buffer treatment actually washes off all the rest of debris. Okay, and this debris like depend upon what kind of sample you sourced out from, so that will be depend upon
What is the composition of
times-
wash buffer?
wash buffer. Again, wash buffer will be having whether it is like sort of alcohol, some kind of different grades of alcohol particularly we are going for in the wash buffer, like from slight like 30-50% to that of 100% alcohol in some cases, because alcohol what it does in the 100% concentration, it will like solidified, or say it will make more prominently prominent structure of or crystallize in the
We
simple
can
way
say it is a
to-
disturb hydrogen bonding.
No, it does not crystallize. It precipitates.
It's like in like when we are eluting in the simple way, we call it like crystallized. We simply like solubilize the DNA into water, and in alcohol we crystallize the your nucleic
So
acids.
that's why it become a insoluble in that mother liquor. It come out of the liquid phase.
Yeah, exactly. Because initially how it is, you are seeing it like I have explained with the sugar and solvent and solute concept like that only. So with the alcohol treatment, it will get like crystallized at one place. Okay. So
Rather,
along the
we
side,
should use the term precipitation.
yeah, precipitation.
You can say it is
Precipitation.
That was the much better word for it. Precipitate. So particularly, it will get precipitated, and while it is getting precipitated, we once it got precipitated in the initial phase, afterwards like that particular dot appears over there of your precipitation, and that particularly we call palette over there. So that palette will be furthermore given treatment of like 2-3 times with different grades of your alcohol, and once it is properly dried out, the you see that palette we consider, because we don't know whether it is going to give us ample number of your nucleic acid or not. It will depend upon the presence of the viral material in the given
In this
sample.
case, RNA is the genomic content.
In the case of COVID,
Yes.
so can you state the difference between, which sugar? Like in this case, ribose sugar is present, right?
Okay.
In
In
the case of
RNA we have ribose nucleic. Sorry,
Ribose sugar is there.
is base, and in DNA we have deoxyribose sugar. Yeah, that's the difference, like basic difference in its backbone. So like in the here we have RNA as a nucleic acid. So this nucleic acid particularly got precipitated. We dry it out, and once it is properly dried, then we treat it with the elution buffer. Now, what is elution buffer? Again, elution buffer will have the alteration of, it will be near your what we say soluble pH, where you have like neutral pH. I mean, sorry, neutral pH. So that buffer will allow that precipitated nucleic acid to get solubilized into the medium. And now you have particularly extracted nucleic acid into soluble phase. Now that liquid phase, again you have to do that. Usually we elute this elution buffer in the quantity of around 100 microliters, but sometimes if we get alters, even 50 microliters is fine. So what we do, we try to get 100 microliter. Usually in my diagnostic lab we used to do 100 microliter, and we divide 100 microliter into 50 microliter batch. Again that 50 liter kept as a backup. Yes, and the further this 50 microliter will be transported to the your RT-PCR lab, and there we carry out further testing on it. Okay, so I will go for the RT-PCR diagnostic lab. So at the RT-PCR, what you have to do basically, as I explained in the first place, there are two explanations to the title. One is, or this acronym particularly, one is real-time PCR you call, and another is reverse transcriptase polymerase chain reaction. So as the first term, when it says, or we are expressing it as a real-time PCR, so basically the polymerase chain reaction we observe and get the results in real time when it is happening particularly.
what exactly PCR? Can you explain?
Yeah. So PCR stands for polymerase chain reaction, and it was invented by Carey Mullis in 1938 around. And it is like basic principle is when you treat a DNA material in the given enzyme like polymerase. So DNA polymerase enzyme. So the rapid cooling and heating of this particular DNA or nucleic acids will multiply the copies or increase the volume of the DNA. That is the like basic and broad explanation, and that is called polymerase chain reaction, because it is like chain reaction is happening and polymerase, DNA polymerase enzyme is involved into this. Now when I have to call the other name, coming back to our explanation of RT-PCR lab, the another name when we call it reverse transcriptase polymerase chain reaction PCR. So in that case, this RT, that is reverse transcriptase, is particularly another enzyme which having major role in this particular diagnostic systems. So reverse transcriptase is basically a one enzyme of central dogma of molecular life, which allows synthesis of DNA using RNA as a template.
RNA assisted DNA polymerase.
RNA assisted DNA polymerase is a gene into the virus particularly.
Oh,
Here we
HIV in present
yeah,
that
it produces reverse transcriptase particularly, like it is responsible for that. Now the reverse transcriptase. Now coming back to the diagnostic steps.
What is that RNA assisted DNA polymerase?
So basically, it with the help of RNA, that enzyme create DNA,
cDNA,
cDNA,
complementary DNA is
complementary.
synthesized. So it has another name. You call, in general it is called reverse transcriptase.
In that case, RNA acts as a template
RdRp.
to form
RdRp is more elaborative definition. Like,
reverse transcriptase.
It is not RNA assisted DNA polymerase.
RNA acts as a template where it
But
forms.
that enzyme name is not RNA assisted DNA polymerase. That is a reverse transcriptase.
That is reverse transcriptase.
And you are talking about HIV that works on a reverse transcriptase.
Say such an enzyme used in a RT-PCR. This reverse transcriptase enzyme.
Yeah, but that term is not there.
Different, term is different.
That different.
now in the like protocol point of view, if I have to explain, the elution buffer what we have acquired, which is having our DNA or nucleic acid, extracted nucleic acid. So we have to treat like we are going to take out of it 5 microliter is even enough, or sometime we took even 10 microliters, if at all, like depend upon the protocol you are having, or the kit you are using particularly. So in our lab we usually go on for 5 microliters of the elution buffer, and those we have to add with reverse transcriptase enzyme.
Can you give us a brief about which genes are majorly considered in case of COVID-19 testing?
Okay, so when the kit we were using, it was particularly targeted for N gene, E gene, and RdRp. Now N gene stands for, or which codes for nucleocapsid, and E stands like E codes for the envelope of the your viral particle, and RdRp, just like sir explained, it's RdRp RNA dependent
RNA dependent RNA dependent DNA polymerase.
Yeah, so those genes.
So RdRp is quite common in virus case.
In as Swapnil sir already have given us brief about the like genetic systems, and he already like correctly pointed out 2 types of gene usually we use, structural genes and non-structural. So RdRp is basically non-structural gene, and E and N are usually structural genes.
Okay, and one more addition. RdRp is not found in humans.
Yeah, correct. Specific.
Specific for virus.
It is
It is a
specific
marker that
for the virus.
For viral particles. Yeah, and the reason behind this, the another reason is also that when we get RdRp, like RdRp gives you a very prominent conformation. In case of N and E gene, there might be sometimes you can get like false results as well, because sometimes you just got in the contact of viral particle which haven't, like, it died in the early defense mechanism itself, and the envelope or what the particles, the fragments are like flowing through your like mucal layer or the blood, like something might have happened and it has been arrived. So that is why we use RdRp as a very prominent gene for the detection, like confirmation, right? So in my case it was
RdRp, which is present in the coronavirus, it is similar with all other viruses, or it has some differential configuration as compared to other viruses?
No, everyone will have the different kind of configuration.
Domain wise?
Domain wise it can differ.
Even genomic content?
Yes.
Again, it is a part of evolution?
Yes. So RdRp of COVID will be different from RdRp of HIV.
Sir explained right now. Sir explained that it comes with a particular property as well. Like in his virus,
That proofreading activity was lost.
and in the COVID it has.
Proofreading loss of.
So like that property, I don't know much of. Today itself in his podcast I got so much of detail of virology, or else I was not having that much of differentiation. Okay, so in this case, what we are using, these were the genomic sequences which were.
So I was thinking about the location of COVID-19 testing is the nasal and oral, you can call it cavity.
Yeah.
Okay.
Nasopharyngeal
Cavity and osopharyngeal cavity, correct?
Yeah. Oral.
Oropharyngeal. So considering the location was targeted quite in this case, because if you consider blood, there are chances that see, our body is having different type of viral infections.
Yes, yes, yes.
Okay, we can have different, and again, kind of influenza virus, we do not have vaccine. So again, that inflation is common for us, and that genes are also present in us. So considering the location and the testing that has been performed with RdRp, blood has not selected because blood is the primary thing that is considered for the testing. But in this case, blood is completely neglected, even it is not considered.
Yes.
Because the RdRp, that is infection of coronavirus, it is particularly in the oropharyngeal and the nasopharyngeal region. So they have targeted the location, they have taken out their home kind of thing, so taken out from home, and deliberately or kind of intentionally has targeted the RdRp gene. So RdRp and the location was the main target, I think, in this case.
Yes.
Is it correct?
The infection will be in the beginning. It will be in the upper,
And that is the reason serum or blood has been neglected in this case.
and later on it will go to the lower respiratory tract.
Yeah, but again, corona will also come in the blood, but they do not want blood sample to be considered as a testing sample.
Yes, yes.
So location and RdRp were quite focused things in this case.
In my point of view, if you may allow, like in
Because you said E and N gene also some kind of trouble is there.
They might have the false positive result.
troublesome for other tests, like I have already mentioned.
Yes,
serum-based
yes,
or
antigen based.
antigen based. So there it becomes like many time it gives like it is very quick, but it there are high chances that you will get a false result.
False result.
So that was very risky thing.
So what are the roles of N gene and E gene?
That codes for codes for nucleocapsid, outer layer of it, and envelope is coating of your genetic materials. I think.
So they are also peculiar in like in coronavirus or also in other viruses, let's say in
Nucleocapsids can have means there are again the nucleocapsid and envelope. It is also the characteristics of other RNA viruses also, but again the specific sequence matter. But in the case of coronaviridae, there are chances it can give false positive. So within the coronaviridae family, it can show the results to other viruses, but from the coronaviridae family.
Okay. So that's why we have to focus on RdRp, right?
RdRp.
To target specifically COVID.
And in addition to his point, also there is another clinical point of view as well. So clinically, when you have like it is a pandemic, so you have very large number of patient or those who are coming to you for diagnostic and testing. So testing numbers are very high. Like in our case, it was 50,000 per day. So the processing is so high. So in that case, having a blood sample would have been like by cost or by time, in all the matter it was like very like having extreme drawbacks in case of, I guess, like in my point of view, to have blood as a sample.
No, that's not a kind of thing. See, again, we are having testing for HIV through serum.
Yes,
So again, it is risky, but still we do with blood.
yeah, but
But
number
quantity,
of I'm saying about the
It's not feasible.
samples coming for the testing particularly, or you are having patients, the testing coming for the HIVs are not
much
yeah
less.
No, I'm talking about the risk. I'm talking about the risk.
risk is definitely we consider.
I think targets are very important in this case.
Like choosing particularly swab through your mucopharyngeal cavity, the another strategy behind it was this.
Viral load, viral home, and the RdRp that is specific for the virus.
Yes.
That's coronavirus. You can say.
The like choices of these genes are like variable depending upon different kits. Different companies used to design different
But RdRp was quite
RdRp was
Specific.
like, quite
common in all the
consistent
kits.
in the most of the kits.
Can you explain about when COVID virus infects your body? How does it, like which receptor it goes to bind and how it acts inside the body? Like there's one thing RBD. So can you just explain about it more? Receptor binding domain.
Okay, so when you like when COVID is getting out of its already infected body, it usually chooses the path of your, as I already explained you about the mucal layer, and from mucal layer, when it blocks the bronchial cavity, it tends to cuff and throw the particles of your mucus in the like aerial version or aerial area. So that particle when flows through the impact area, and it goes, and a healthy person when inhales those particles, and that particle goes inside his cavity and crossing all his primary defenses. If it successfully lands on your oropharyngeal cavity, through where because of fluidity of your buccal layer, it gone in your um lung cavity. So there particularly it infects the, yeah.
No, I mean, I
So
would
there,
like to know about,
there is ACE2 receptor. So angiotensin converting
converting enzyme.
converting enzyme. So
So
that
yeah,
is the
I wanted to
receptor.
know about that thing, like how exactly what
That is the receptor, and apart from this, there is a one protease, that furin. So what it does, it cleaves the some kind of glycoprotein sequence from there. So now the fusion peptides will open, and those fusion peptides will attach to the membrane.
What is meant by fusion peptide?
They are hydrophobic amino acid sequence, small sequences. So
Who creates these fusion peptides?
it is inside the what you can say.
Coronavirus.
It is inside. No, it is inside the glycoprotein or inside the spike protein.
Spike protein of coronavirus.
COVID virus, yeah,
So
COVID
it is
virus.
present on the coronavirus. It is created by coronavirus.
Yes, yes.
Means entry particularly takes place like the, what we say, general method is like any virus, like it has a receptor like enzyme and substrate lock and key hypothesis, like that. The binds, your receptor will bind with a particular protein present on the
So that
Okay,
is RBD,
just-
right, sir? Receptor binding domain.
Yes, yes. One more addition I would like to do, and that's why people were considering it as what artificially created virus, or what lab born virus, lab
Cultivated virus.
Do you think is it possible to create artificially engineered virus?
No, not possible. But in case of COVID, I would like to explain. The specific mutation wasn't there before. So maybe after. So basically, it is COVID is bat reservoir. Means, so
Reservoir of what?
reservoir, it stays inside the bats. So bats are actual
In the bats.
bats. So
Natural host is bat.
natural host is bat, and somehow from the bats there are the intermediate hosts like pangolins and other ferrets, civets. So during that might have it changed. Specifically, that mutations have been created.
Which mutation?
That furin spike protein and cleavage of that furin cleavage protease. So it wasn't before, and that's why it wasn't infectious before. But somehow, what we found the sequences which exactly match, or that mutations was found in the pangolins. So in
That mutation, that regarding the interaction of fusion proteins with something, that is observed in case of pangolins.
So somehow it has mutated inside the pangolins, and then it started.
But it wasn't that much virulent as compared to humans to pangolins.
No, it wasn't.
It became pandemic for us because it wasn't identified our body to deal with the coronavirus as it dealt with the pangolins.
And that specific mutation has caused to make easy for the virus to infect.
I have one question. During the transition period, virus decided so its next host.
I don't think so. It decides it.
I don't think so.
So right now, coronavirus, it is in the bats, it is in the pangolins, it is in the humans, and all other intermediates.
Intermediates.
Even in some of the animals like cats.
Even tigers also.
We were unfortunate that we had pandemic with it. All other weren't the part of pandemic system.
Yeah.
Yes. And basically, due to that specific mutation.
That occurred in the pangolins.
So it does not happen same with other coronaviridae members, right? Like before MERS and those.
No, MERS and SARS, they have the different receptors system, I guess. But in that specifically SARS-2 case,
COVID-2.
that particularly that mutation make furin to cleave that part spike. I don't know even it is spike protein or other glycoprotein, but it opened the fusion peptides, and that make possible of
So route of infection was quite dissimilar as compared to the earlier ones,
yes, yes,
and that became the reason our body did not fight against our body did not fight well against the COVID virus.
yes. That is also the reason, because it is first time, so we don't have kind of memory regarding.
Okay,
And the severity is also like increased because of it.
okay. So let's move on to RT-PCR.
Yeah. So the sample is in the treatment under reverse transcriptase. We have added reverse transcriptase to our elution buffer, what we have, and you prepare like series of wells. And now what is wells is like a small type of appendrof tube itself, but it is like the, as I'm already saying, we have a very small microliter level of quantity to deal with, so that is why it is very small.
Microwells.
Yeah, micro. So we for the sake of your polymerase chain reaction, it has been like made. So like in general manual way, we have the normal we use the normal appendrop itself, and the machineries what we observe today actually have like very broad and elongated setup. Like you have sample like loading, then different levels of thermal treatments to it, like what we call thermal cycles in it. So basically, before going to that, once you when you treat your elution buffer with the reverse transcriptor, what is happening at the molecular level? The this RNA, which was the nucleic acid nucleic material of your viruses, is synthesizes we say complementary DNA. So the single stranded RNA is getting converted into, or a copy is created, a copy of complementary DNA is converted with the help of your reverse transcriptase. Now this complementary DNA we are going to amplify using polymerase chain reaction. Here the polymerase works to amplify the amplify the number of complementary DNA in this system. Now the particular steps here we like, as in this earlier introduction itself I said that cooling and heating of the DNA like molecules in the particular this medium, along with supplied with required DNTPs and enzymes, it will increase its volume. So that was the basic principle of your polymerase chain reaction. So in this particular given setup, we have like improvised machineries which is very compact, and there you can do it. The thermal cycling will be done in front of you, like one after another. Rapid cooling and rapid heating systems are like already inside, embedded inside it. And due to the fantastic detection systems using chromophore particularly, you have ability right now to observe this increasing viral load or nucleic acid material in real time. That is why it also called real time PCR. Okay, so these steps, particularly heating and cooling, what I am telling, so it is uh, figured out like we call first step as a denaturing, then we have annealing, and then we have extension. Okay, so denaturing takes place at the 95 degrees Celsius approximately, like around it. Then we have annealing at 50 to 60 degrees Celsius approximately, and then we have we do the extension at the 72 degrees Celsius approximately. So these are the steps which we follow in your polymerase chain reaction, and this particularly called as a one cycle thermocycle. That is why it is termed as a thermocycle. So once when you follow one cycle, you got the like one copy, you got another copy, those two will go for next cycle, it will multiply, the next cycle it will multiply. So accordingly the cycle you carry for further, the amount of your DNA material or whatever the nucleic acid material will get multiplied, right? In this case, like always here in the PCR, in the case of PCR particularly, it is always DNA, because we are having cDNA from the RNA, and in the normal case we have DNA itself. So DNA will go on multiplying, and the nucleic acid material will increase in its copies. So how we are going to identify it? For that, I have to explain you the exact what we say process of replication here. So during the replication, you form the protein which associate like directs to your particular origin of your replication, where we call two forks are formed, we call origin of replication, and there you get attached with DNA polymerase, and DNA polymerase will with the help of first what we RNA primer, it will start 5 prime to 3 prime synthesis of this nuclear strand. Okay, this is called leading strand, and the other strand is called lagging strand, where the synthesis of DNA will take place in the fragments, and that's this fragments is called Okazaki
Okazaki
fragment. That's our
fragments.
basic of our replication. All right. So in this case, we have DNA polymerase. It has been entered, and for the primer, what we have introduced in this case, particularly, I will be talk about very simplest probe which we use. That is tech probe. So what it consists of? So the primer is designed for the targeted gene, and you have one quencher at one point, and another point you have chromophore. The chromophore is basically molecule which gives out particular wavelength. Now the particular wavelength, when we say, so different wavelength will also related with the different color. Okay, so in some cases they uses UV rays as a source of light to get this color. In some cases they use like in the current modification, they uses laser particularly to get the different light. Now what happened when primer sits at that position, and the particular primer has been like found its place, and it got aligned. So the first thing what happened is, before starting aligning the next DNT piece in the sequence to lead the strand, it will cleave this chromophore, right? So as soon as chromophore goes away from your quencher, the property of a quencher is it blocks the like light emission of your probe. So as soon as you get out or away from your quencher, so it will start to like illuminate in the provided light. Now probe will continue its synthesizing of particular gene. So that is how the strand is formed, and you got the first copy.
What is quenching?
So quenching, as much I am understanding, so quenching is basically removing, or you have this chromophore which giving you particularly signals, right, in the form of other excited like electron in the form of light. So quencher basically traps this, or absorbs this energy or this light coming out. So you are like this light will not get detected by the detectors, so it remains submerged. So when, as soon as the chromophore get cleaved away from that your probe, so after that it gets away from the quencher, so it will give you the light, and that light will be sensed in the detectors. That's how you track, like live real-time tracking has been done of your this uh, DNA replication. And in the term replication is making multiple copies of the
Amplification.
sorry
Amplification.
yeah, amplification particularly. So this is how your copies of your DNA is made. So the how we are detecting. So I will come to the detection part. Now in the detection, the basic principle is number of cycles, that is termed as a CT value. So number of cycles, the higher the volume of your viral load, lower
So
the
the
cycle,
cycles,
and lower the your uh, viral load, higher
higher the
the cycle.
cycles.
Why it required? So let's imagine it is happening like first viral load you have detected. You have let's say in one sample you have one viral gene, and in other you have 15 or 20 genes. Okay, so the more amount, like in less cycle, you will get more amount of signals in the higher viral load sample, right? And in same amount of cycles, you are getting like very low signal in the uh, low viral load sample. Okay, so we like set as per the your calibration has been done by the test kit providing companies. So there is certain value that up to that the city will be carry forward, and if that threshold is not crossed by the, or you haven't detected, so it will be considered as a negative. So it will give you the peaks on the detector systems. Particularly it is giving as a graphical representation, different color, like that representation. The point over here is we usually have uh, identified, like in some kits we have 7 15 city values, or in some, like in ideal or in average we have seen is 13 cycle as a positive we have detected, and the result usually given as a positive, negative or non-conclusive. In case of non-conclusive, that there must be problem with either your samples, there might be contamination, there might be any other like detection part of interference might have happened. So in that case the clinical whoever the authority is there, so usually they prefer to uh, get it done by the result samples. If the result samples continue to do that, so we have to do this whole process entirely again. So that was the interpretation. Once you got positive, negative or inconclusive, based upon that the report, printable report is generated. But during the COVID, due to huge number of sampling of handle, we usually share this as electronically, and also it is very helpful to avoid the like taking contamination to the outside of your lab. So that was the thing, and based upon your reports, now we come to the third facility of this whole pandemic response, we could call, that is the quarantine. So based upon your report, if at all one person is admitted as a symptomatic patient, so he will be, according to your, whether you are positive or negative, accordingly you will be provided the facility of quarantine systematic. Like we generated one quarantine system where the patient is kept away from the rest of the patient or rest of the normal people, and he was supplied by all the required amenities like food and whatever the possible things and care he might need it. So according to that the facility, so what it like impacted. So as it is very, until unless you don't develop a particular vaccine against the COVID, you do not have any like a defensive mechanism as such, apart from having like normal defensive mechanism, rising your temperature, having mucal layer, increased mucal layer, so you could throw it out, that's it. Or in the extreme condition you come to the like level of interference get increased, and that generates like swelling in the whole systematics, like these things used to happen. So in that case you will require to have a ventilator and oxygen systematics, or else it is not required as such. But that apart from that defense mechanism, you will be like, at that point we were not having any mechanism to deal with it. So this was the only way you can contain the spread of virus. That was the only target at that time in front of the systematics. So lesser the infection, like lesser the spread of infection, like what scientifically idea behind it is, lesser the spread of infection, lower the chances of mutation which could occur to these viruses, and there might be lesser chances to get any other spread or any other.
No, actually virus is having its own evolution timeline. So whether it is infecting, not infecting, it is finding the host.
Yeah,
So whether it infects to us, it does not matter.
no, they spread into the population. The more host it will jump, there are more chances that it will have to develop.
No, again differentiality is there. If it is transitioning, I think rate of evolution will be higher. If it's targeting certain community, again community is the same. Immunological response is quite similar. So that is the reason after pandemic, when we vaccinated, we do not have infection of coronavirus again, because again if it shows mutation, still our body has memory against it. If it changes the category and again reinfects us, I think that time
Already have memory.
Yes, and one more thing, like viruses can lose their virulence once.
we all are in the survival mode. As you mentioned, it is transferred from bats to pangolins, and then pangolins to intermediate, some stages are there, and then humans. So corona is with us for a very long period in the back. But once it is infected to pangolins and the mutation that has occurred, that has eventually resulted in our pandemics. If it wasn't in the transition phase, it was in the bats, and the community of bats was in the larger percentage, it wouldn't have considered as a host for us. So mutation, I think it depends on the category that is it is infecting.
Why do SpO2 level fall in the case of COVID-19?
Where?
SpO2 level falls,
SPO.
like blood oxygen level.
Okay, blood
The
oxygen.
target organ is lungs.
That is the cascade mechanism. That is the cascade.
The infection of virus results into swelling of your internal like lungs tissue particularly. Now those lungs tissue which we like alveoli,
Alveoli.
yeah. So there what happens, the swelling leads to the mechanism. What it was expected to do is inhale and exhale, and do at that point you do the
Gases
exchange of
exchange.
gases, yeah, with blood. So due to your swelling particularly, the movement or this particular role of whatever the role nature has given to it, it usually fails to do that, and because of that it always like this creates another wave of signaling in your body. Once you got that, you are trying to do the inhaling of air more and more to dilute or take inside the more and more oxygen. So because of that, it create pressure on your lungs, and because the alveoli are incapable in the swelling condition, so it also creates the pressure on your heart. So it like a cascade of mechanism happens. So to avoid like if some are already having chronic diseases like exhibiting heart disease or something, so in that case this pressure which has been formed, there might be chances that the heart failure, or we have seen that organ failure or multiple organ failure can be like have observed already in the patients. So the first thing a doctor used to do is to check the level of your oxygen. So that shows, and like it is directly proportional to your capacity of lung to like do the exchange of, do its role of exchange of gases. So SpO2 of your blood directly indicating is the role that has been interfered, and it is like whether it is going to be critical or not, and accordingly the decision of like to put on ventilator will be the decision of ventilation, or ventilator should be provided or not will be taken.
Okay, so that pneumocytes are there. So on their surface there are surfactants, which are made up of lipids and proteins. So after infection they are removed. So that's why the cells or alveolar sacs become rigid. So it is hard to pump or take the breathing in and out. So that is also reason we.
And I guess because of that might be the like body is also trying to create a mucus, like our
No,
body is particularly like what I want to understand in this thing is like as a virology point of view, I do not understand the body's forming of mucus is particularly as a defense to trap the viruses, or it is already like infected to your cells. So in response, it want to like get that like in other cases how immunity works, that it supplies this glucose and like the infection should go away. So like that, I do not understand the role of like why mucus
yes, increasing mucus is general kind of
response.
response for the to trap the virus or pathogen.
Exactly.
That is
innate immunity.
yes.
Part of it. Okay, so like that I wanted to clarify that itself, because from the start we were discussing oropharyngeal sampling. So okay, my idea is
But as he was concerning, in mucus there will be less
viral load, and yeah, the expectation of cellular
Virus is very smart. He knows what to do. That virus knows what to do. So considering that mucosal as a defense system, virus already knows about it. Because if you talk about adaptability, it is very smart as compared to us. So maybe lungs is the proper choice for coronavirus to make
Yeah,
a
that is
home.
true.
there are more ACE2 receptors on the
So
lungs.
initial infection may be in the nasopharyngeal and oropharyngeal region, but later on the actual home for the coronavirus is the lungs.
Yes.
That's what I feel. Where there is a less, maybe I don't know exactly, but less mucosal layers as compared to the oral region and the nasal region.
That also helps the transfer of
Again, it is advantages for them also to be there in that region.
Yes.
It's
Maybe
kind of advantage and disadvantage.
it's both, and
It is strategy kind of. Both are having strategies for each other.
each other.
also in children the infection was less, and that was the reason that AC2 receptors were less in young children.
It's more in young population.
In adults,
It's
and
less.
that's why.
So, please, sir, see, viral genomes are having very less nucleotides. So, what don't you think that there is possibility of creating engineered viruses?
Yes, it is hard to create the engineered a virus, because even if I say, suppose I want to make a artificial virus or engineered virus, and I will choose extremely highly virulent like Ebola, Marburg, or suppose COVID. So even I try to put them as a clone, intact, as an intact total virus genome. It is hard to predict whether it will grow inside the cells.
Okay, as you mentioned that if I transfer the genome, I don't know what's gonna happen inside the cell.
And suppose even if it is replicated inside the animal cell line, it is hard to predict whether it will replicate in the human or animal bodies or not. So it is very hard to find out which combination will work. So for the viruses, they themselves know which is the best combination to infect rather than us.
So the activation is also like dependent of response of host, or it is moreover like the host will uh, treat, or host is like play a crucial role into the survival or activation, like in this case if we are engineering particularly. So in that case, in my point of view, activation of that virus will be like when you are doing even that kind of thing in a way, so you had to be enough smart that like host the target, like because of that only in possibility of biological warfare, once they like predicted that there might be like in the future you could also have uh, personal, like person targeted pathogens you can engineer.
Bioweapon,
So yeah,
Like
So
Pseudomonas, right,
person,
sir?
specific person.
if I spread this
not a
pathogen,
person, community.
no,
He
no,
said
even
person,
specific, like
single person.
single person's GNA specification I could like take out possibility whole if I get your whole genome sequences. So just like you identify
But why would
RdRp
they
very
do that?
specifically, huh?
Why would they do
If
that?
they want to target specific people like
Because
VIPs.
that like bio war, if it is
It's quite difficult to understand.
It is difficult to find out the individual
If we
person.
assume that we are creating a person, it's quite
Yeah.
long ahead journey that we need to do.
Theoretically, you can create, but it is hard to,
In your mind, you can create.
because even we consider whole human community to find out the best virus or to design the best virus to infect. It is hardly possible. So inside that also you want to target specific person. So it is one more layer of difficulty is there.
Santosh sir, you have worked in the area of material science for a while now. You have synthesized different materials like silver nanoparticles, again zeolites and magnetic nanoparticle. So could you give us a detailed overview of your materials that you have been synthesized during your course of investigation, starting from the silver nanoparticles?
Thank you, Shubham, to being me here. Uh, I started my journey in 2015. That was actually I don't know anything about nanoparticle, how it's synthesized and how the reaction works. Being a chemical teacher, being a chemistry teacher, I observed lots of reaction. I'm very much fascinating about precipitation. So nanoparticle is somewhat synthesis of nanoparticle is somewhat kind of precipitation reaction. So my journey begins with a silver nanoparticle. I synthesize silver nanoparticle with the help of gardenia. a gardenia plant extract, methylenic extract. So I found that
What are the different methods to synthesize silver nanoparticles?
there are various methods which we can synthesize silver nanoparticle, like physical, chemical, and biological.
What are their principles?
Physically means there are actually all methods fall into two types: bottom up and bottom down approach. So physical methods, we can use uh, metal as a part, where we like lithography, we can synthesize, we just make smaller particle, and also we can use ball mill to synthesize smaller particle, where at some time there's a formation of nanoparticle level. Okay, in case of chemical, we use chemical precursor um, as a starting material. Like in my case, silver nitrate as a it act as a precursor, and which is reduced by various reducing agent like sodium borotate, and with the help of capping agent, we prevent agglomeration at a certain level, where nanoparticle present in the mother liquor. In case of biological, it is the same method like a chemical method, but in that case we use plant extract, where silver precursor, in my case, uh, precursor get reduced, and it's stabilized by biomolecule present in the plant extract.
Why plant extracts are targeted in this case?
Because it is a simple, non-expensive, and actually we there is a no any other chemicals required, which is maybe produces harmful byproducts. In green synthesis, we just get plant extract and precursor both.
So when you use plant extract as a material along with your silver nanoparticle precursor, it is green synthesis.
Yes.
What are the principles of green synthesis?
It should be inexpensive, and it should be uh,
Do you think use of plant extract reduces the toxicity of material?
okay. Yes, why not?
How?
Because in chemical, if you compare the chemical reaction and green synthesis, in chemical reaction we use various binding agent or capping agent, which is harmful to the environment.
What is the difference between binding and capping agent?
Capping agent, we can say,
What is
particularly,
capping agent?
which arrests the agglomeration of nanoparticle, agglomeration of uh, the when there is a precipitation, or to understand what is the capping, we have to understand what is mean by how the synthesis form, how the nanoparticle synthesis.
Go ahead.
During the synthesis, or any synthesis in chemical and green synthesis, there is a formation of actually there is a supersaturation, where
What is
when
difference between saturation and supersaturation?
in supersaturation, uh, the uh, it goes in uh, the precursor, or after the conversion of into the reducer form, the equilibrium between soluble to insoluble, it shifted to insoluble part. So there is a there is a there is a high amount of uh, that particular entity goes to the beyond saturation, so it becomes super saturation.
Who reduces whom in this case?
In this case, that precursor get reduced by a reducing agent. In chemistry, it is a sodium borotate or maybe citrate ion. It can reduces that nanoparticle, that precursor.
In case of plant extract?
In case of plant material, actually plant material itself, it contains various chemical, thousands of chemicals there. Maybe there is glucose is present, amino acid also there. So maybe it act as a reducing agent.
Okay. Any specific category of plant extract that is targeted towards the targeted or considered towards the reducing agent?
Every plant extract has actually basic chemicals, like it contains glucose, it contain amino acid, it contain carbohydrate.
So utilizing a specific category of plant is not the primary concern. Just use any plant extract.
No, specific plant is very important.
So composition of plant extract does matter.
Yes, composition from plant extract as well as the
So that is why I'm asking which category of plant extract is more dominant in case of reduction process.
Suppose if there is a like gallic acid or polyphenolic compound, if any plant extract contain more polyphenolic compound, shows more reducing power. So polyphenolic compound act as a powerful reducing agent
Why?
because it contain it shows very actually it reduces easily plant.
Take an example of silver nanoparticles and tell the exact reaction what is reducing.
In case of synthesis of silver nanoparticle, silver acted as a precursor.
Okay.
The plant extract which contains carbohydrate, amino acid, and various.
So silver nanoparticles are directly added into the plant extract.
Yes.
Okay, and there is a reduction process. How?
Yes. Actually, the plant material it contains carbohydrate and protein, amino acid, which actually basically it is a chemicals.
So AgNO3, once it is dissolved in the water, what it produces?
It produces silver ion.
Silver ion.
Silver ion, Ag plus.
Ag plus, okay.
It get reduced into silver metal.
So Ag plus is having 1.
Ag 0. I can say it is a Ag plus. Silver atom is get reduced into the silver metal. Silver Ag 0. The charge on the silver atom becomes 0, and due to the intra-atomic interaction, they get agglomerated to form a cluster. That cluster.
Okay, wait. When Ag plus is converted to Ag0, it means that Ag plus is having a lost electron that has been accepted from the plant extract and converted into Ag0.
Yes.
Okay, so Ag0 at this moment is having stabilized form, that is elemental form.
Yes.
Okay, so in the elemental form you're gonna use it for your application.
Yes.
So Ag0 at this case is having electron from the plant extract and more specifically from the polyphenols. Okay, so polyphenols have provided hydrogen to Ag plus, electron to Ag plus.
Actually, there is a transfer of electrons.
Electron from polyphenol to Ag plus ions. Okay.
So after there is a formation of clusters. Cluster means there is
so Ag0 come together and form a cluster.
Yes.
Why?
Cluster means the some atoms of silver 0 come together. This is due to the intra-atomic interaction. It is
intra-atomic interaction.
Intra
or inter-atomic interactions?
Inter-atomic interaction.
Why there is interaction? See, elemental form is stabilized form. Why they are interacting with each other?
There is an interaction between it is a maybe electron repulsion, somehow electrostatic interaction.
Okay, wait. When we see the atomic configuration of silver having one added electron from the polyphenols, so electrons which has been added from the polyphenols is giving stability in a way as compared to Ag plus.
Ag plus is a soluble part. Okay, we are considering cluster which is not soluble into the mother.
Wait, we will go to the cluster. Right now Ag0 is there, and Ag0 it is having stabilized form, it is not having any charge, it is neutral. Okay, and it is interacting with another neutral form of that silver.
It is a kind of electrostatic interaction.
No, wait, if there is okay, that's fine, electrostatic interaction is there, but again nucleus interacts with electron, that is the electrons which are moving around the nucleus. Again there is a electrostatic force of attraction. One electrostatic force of attraction attracting the other electrostatic force of attraction, it means that there is a sharing of electron in between them. It means that the electrons which has been taken from the polyphenols having a different energy level.
That is not case.
No, see, if clusters are attracting each other, it means that the cluster has created electrostatic interaction in between them. That is the electron sharing from one orbit to another orbit of AgNP. It means that
There is no any chemical bond formed between the cluster.
that is what I am telling. There is no chemical bond. That is the reason I think it is because of the discrete energy level
I don't know, but
of electron that has been taken from the polyphenols.
no, it is due to the actually what happened. I think after formation of Ag0, so there is a solubility decreases into the mother liquor of Ag0. So Ag0
Solubility decreases, it is because it is converting into the elemental form.
Yes.
That is the best reason. One elemental form connecting with another elemental form, it is not as same as what we see in the real world, that metallic form. It is quite different metallic form that has been generated because of Ag0.
Yes.
Okay, and one Ag0 is interacting with another Ag0. It is because of the electrostatic force of attraction, that is quite obvious. But the electrostatic force that has been generated in between them, it is because of the electron that has been taken from the polyphenols that has been shared between them. And now clusters are attracting with each other, I think, because it has created around a kind of structure which is sharing the electrons to one another to make stable structure.
Okay, so it is a basic of cluster formation. Some atoms to get together to form.
See, you told me there is a stopping, that is capping reaction is there. So you need a capping process. Okay, addition of some, you said citric
But
acid.
it happens after some time during the formation of clusters.
No, you are saying there is agglomeration.
Yes,
Agglomeration.
that I want to say that many clusters come together. There's a actually there is a probability. Always there is a probability. We cannot say it like a straightforward reaction.
No, see, right
There
here,
is
clusters are attracting.
capping of cluster is also there. Many cluster get incorporated. There's a function of big.
I accept. Whole reaction is happening inside a beaker where there is water, there is polyphenols, and there are silver nanoparticles. Right here, the rate of conversion of Ag plus to Ag0, it is quite continuous. You don't know how there is change of Ag plus to Ag0. Again, there is a rate, certain rate.
Yes.
So Ag0, which has been converted from Ag plus, taking electron from the polyphenols, now they are attracting each other because of the elemental form they have been created. See, electrostatic force of attraction is there in between them, no doubt. But how? I think because of sharing of that electron, which is generated from the polyphenols, they're taken. Again, the electron they have taken, I think it is having a discrete energy, and that energy is responsible for connect. Okay, it connects with quantum. It connects with quantum where quantum confinement comes. What I'm telling is the quantum confinement. That is the stabilization of a structure because of the quantum confinement that is generated due to discrete level of energy between the electrons. And that has resulted in providing a certain kind of energy, discrete level of energy to whole cluster. So clusters will attract another cluster until you do not stop the reaction.
Somewhat.
It happens in quantum confinement actually.
After formation of that cluster, actually there is a probability. Okay, some cluster get together form big part.
Agglomeration.
We can say it is agglomeration.
I'm
telling
this because I have worked on
We cannot stop agglomeration. We just prevent some after some
No, you are stabilizing the discrete level that has been generated
with the help of capping agent.
with the help of capping agent.
So after some time, there is a probability. Some clusters get on their surface the capping agent. Some cluster would together form big particle in the nanoparticle range, and there's a formation of capping. There's a capping agent like say we can say it is a polymer type. So
In your case, what was the capping agent?
in my case, capping agent might be a uh, starch. Starch also acts as a capping agent.
So you use starch as a capping agent to stop the reaction.
There is a starch.
How was the structure of silver nanoparticles in SAM structure?
In my case, actually, I used a methylenic extract. So it is unusual.
No, SCM
I
analysis.
found a triangular shape.
Triangular shape agglomerated with each other.
No.
There wasn't any clusters.
There is a cluster actually, but I found that there is a triangular shape,
Do you
which is
think
unusual, and maybe it is. I think maybe it is due to the which kind of solvent we use during the synthesis, because it very helpful to design the shape, or because dielectric constant, the polarity of the solvent, which kind of system is there. So it
okay, single nanoparticles mean cluster of a is not
many atoms get together form there is a nano level particle,
Can you
which is not soluble in a mother liquor.
can you change the shape of the nanoparticle based on adjusting the configuration of your
So yes, solid system and pH of the solution, which kind of so it is a kind of art. Synthesis is a kind of art.
have you tried different, like, let's say, optimizing these shapes of your nanoparticles?
I used it, but I actually not published that data.
Okay. So which kind of shapes it did generate? I mean, changing the configuration of your linker molecules.
Triangular.
And
what is a liquor mixture?
Huh?
What is a liquor mixture?
Mother liquor. Mother liquor means the reaction mixture. In chemistry, we can say it is a mother liquor.
AgNO3 with plant extract, that is mother.
Any reaction mixture known as a mother liquor.
Okay, and then further what is that? Means from mother liquor you gonna transfer somewhere it? What is that called?
Suppose any reaction taking place, so in chemistry we say it is a mother liquor, because it are any.
Actually, homeopathy uses this terminology more often. That is, mother liquor means actual extract, which is saturated one, and then when you dilute it, that becomes the actual sample that has been provided to you. So the parent solution that is with saturated, no dilution, that's a mother liquor.
Okay. One question I have, like, when you are we are using plant material, right? And as you are we are discussing here interference of phenolic content of plant extract particularly. So even pigment might get interfere, right? So is really if at all that is the, like you said, yes. So the sourcing from which plant part particularly we are taking,
Everything is matter here.
Yeah. Okay.
Because we cannot predict if suppose same plant but different solvent system, so there is a different result. So we cannot predict which kind of result we found.
And different part also give you
Different results.
in your experiment, which part did you take?
Because every part of the plant shows different chemical compositions, so that's important. Also, which kind of solvent system is there, which kind of temperature is there, which kind of pH is there, which kind of precursor is there. It also affect the product, the main product of the or any nanoparticle. We cannot predict.
In your case, which part did you use?
Leaf.
Leaf extract.
And last question will be in this area. So just I want to know what would be the general application
Nanoparticle.
we are if in your experiment you are expecting where to apply it, sort of thing.
After synthesis, I use that nanoparticle for the antimicrobial activity.
Okay.
And degradation of dye with the help of sodium borate.
Before application part, did you check the toxicity of your nanoparticles?
No.
It is very crucial when you apply.
Yes, exactly. Because silver is known to produce ROS, reactor oxygen species. But in my case, I'm not tested for the animal cell. Many applications says it is used for wound healing. But in my case, I'm not studied that aspect, but it is very important.
So let's move on to the application part. What were your applications?
There are two types of application. First, there was two kind of application. There were two applications I studied. That one is antimicrobial activity, and second one is a degradation of 4-nitrophenol with the help of sodium borate.
Can you explain about antimicrobial activity? Like which microbes did you use for your antimicrobials?
Multidisciplinary bacteria. 3 or 4 bacteria was there, and it shows. Actually, I tested that. I use SEM image, and I found that that bacteria get agglomerated after treatment of silver nitrate, a silver nanoparticle. So it is due to the it's stuck on the bacterial cell wall, and there's a leaching of silver ion into the that bacteria. It induces ROS, and thus after due to the heavy load of ROS, eventually the bacteria dies.
How silver nanoparticle produces ROS?
Uh, Actually, it interact with the H2O2. So there is a cycle of chemical reaction which induces free radicals, and due to the H2O2 present in this bacterial cell. So eventually it induces a radical and ultimately damage to the lipids as well as the DNA present in the bacteria, and thus the bacteria eventually dies.
Can you call it as AOPs, advanced oxidation processes?
Um, yes, we cannot. We can say AOPs.
Because AOPs basically produce free radicals.
Yes.
Like when combined with UV light, or let's say ozone.
No,
I mean, H2O2 combined with UV light.
we can say it is a AOPs.
Yeah, so it basically forms free radicals.
Fenton kind of reaction, na?
Yeah. Phantom person is a different thing. Phantom uses Fe like
हाँ.
like Fe ions in order to produce free radicals. I mean, phantom person is different. AOPs are different.
Let's talk about your second application.
Now, degradation of 4-nitrophenol. It is a dye, toxic dye. In presence of sodium borohydrate, that silver nanoparticle act as a catalyst. It act as a absorbent for both borohydrate ion as well as 4-nitrophenol. So that both entity come together in presence of that silver nanoparticle, and there is a sharing of electron, and that 4-nitrophenol become a 4-aminophenol. That is a non-toxic product.
Who shares electron to whom?
4-nitrophenol uh, get reduced to 4-aminophenol. So in that there is a transfer of hydrogen to the 4-nitrophenol.
Who transfers the hydrogen?
Actually, silver nanoparticle act as a mediator. So
Okay.
it mediate the transfer of hydrogen to the 4-nitrophenol. So it become a 4-aminophenol, and there's a formation of leucobase. So the colorless product will be formed.
Leucoform.
Leucoform.
So aminophenol has a leucoform.
4-aminophenol is colorless in nature. 4-nitrophenol is a, I think, yellow color. So disappearance of yellow color after addition of silver nanoparticle.
There is category of paper, or you can say that there are number of papers who without going for HPLC and mass spectroscopy claim about the degradation of dyes. Like you mentioned about the 4-nitrophenol, that is the dye in your case and it is having leucoform. There is a category of dyes like triaryl methane dyes. Example, malachite green, crystal violet. So these dyes are having leucoforms and it is dependent on the pH and temperature.
Yes.
Okay. So in alkaline pH, it changes its form to colorless form and it gets into leucoform. Leucoform is again toxic to environment as similar as malachite green, and even there is a transitionary form that is called carbinol, which is again a colorless form. So without going for knowing the mass spectra or HPLC analysis, claiming that there is a degradation of malachite green through chemical reaction or through any biological system reaction, do you think it is appropriate to do that?
Because there is if you check 4-nitrophenol UV spectra, it is a different, and 4-amino spectra is different. So during the degradation, 4-nitrophenol spectra that the peak becomes reduced, and 4-aminophenol peak become increases. So anyone, there are lots of paper which says there's a formation of 4-aminophenol during the.
Do you say in your paper that it is a decolorization or it is a degradation?
It is a degradation.
How it is degradation? It has changed its color only.
Yes.
So how it is degraded?
So 4-nitrophenol become a 4
Degradation is actual concept where there is a proper mineralization, conversion into CO2, conversion into different forms other than metabolites. That is actual degradation.
Can I add something in this topic? So basically, as you raised that point, so basically dye is made of. I mean, if we see the chemical composition of dye, it has chromophores and oxochromes, right? So chromophores basically they are responsible for giving color. Basically, it absorbs the light. Okay, and in the case of oxochromes, they basically just give the stability to the dye. So in the case of, I mean, as you mentioned, that there are many studies which actually interprets about. I mean, they come to the conclusion about without going for mass spectra and HPLC, and then they conclude that there is a removal of the color, or basically, like as you mentioned, that there is a difference between decoloration and degradation, right? So in the case of UV visible spectroscopy, so what basically UV visible spectroscopy does, I mean, UV light basically absorbs the chromophore color. Okay, I mean, absorbs the light of the chromophore, and it gives the detection based on how much is chromophore is absorbed. Okay, so this is not actually an indication of degradation. So in degradation, I mean, if I would like to explain about degradation process, like in UV visible spectroscopy, UV visible spectroscopy actually it just measures whether the chromophore is it's just broken down, but it's not actually converted into non-toxic forms, or let's say CO2 or water, right? So most of the people actually they just show the data that there is a change of the color, but they show it as degradation, but basically it's not degradation, it's decoloration. So in order to differentiate between that, apart from UV visible spectroscopy, you have to go through two spectroscopy methods, I can say. First is HPLC method, or UHPLC method, where the parent dye molecule, I mean parent compound, is basically resolved, and it separates the parent dye, parent molecule from intermediates, so you can identify how much is parent dye is remaining. Okay, and after going through HPLC, the next step is mass let's say LCMS, so mass spectroscopy. So basically, what it gives, so it gives actual mass features of the intermediate compounds and parent molecules. So you can identify based on the let's say masses of the intermediates that are formed. So basically, it gives exact composition of the masses, whether that particular dye molecule is degraded into its less toxic forms, like let's say CO2 water. So this is the basic mechanism.
So mass spectra helps to know that whether there is production of metabolites or there is a proper mineralization process.
Yes, certainly.
So in your study, did you perform UV visible, or
Basically, we can say it is a chemical transformation.
No.
so in your
When you come in the category of emerging contaminants, you have to be more careful about whether it is adsorption, whether it is sorption, whether it is absorption, whether it is decolorization, whether it is degradation. These are the few terminologies that you can more preferably can use in this context. Chemical transpiration is a very vague concept in this category.
So in your experiment, like as an application, did you perform UV visible, or you also went to HPLC and you?
UV visible.
So it was basically decoloration for you, right?
That paper focused on the synthesis part.
Okay.
Silver nanoparticles, in recent I have seen an ad where silver nanoparticle technology has been used, and it is a patented one. So they have used silver nanoparticles as an antimicrobial agent in a paint. So again, having a question here, we now having a different category of emerging contaminants from different industries, okay, and we are flooded with it. Prominently heavy metals, dyes, cosmetic industries, pharmaceutical industries, and now in the case of paint industries, they are also coming more forward. So having knowledge about that there is dye category which has been contaminating for our water system for a long period, and we are not having an appropriate solution towards it. Coming with an approach where silver particles have been used or embedded or connected with the dye molecules. So the main purpose behind is that to increases durability and sustainability in the nature. So coming with an antimicrobial property, which is a good for us, which is good for the commercial market, but increasing its sustainability in the environment, that is particularly of dye molecules, do you think it is appropriate or somewhere we are taking a wrong step?
Actually, after we actually don't know. After some time, if you spend lots of time to get various scientific data, based on that data, we can predict that. But now we don't know.
As you mentioned, silver nanoparticles when interact with the bacterial system, they
Yes.
produce
But
H2O2
also-
and there is ROS production. So again, when you use that pent, it is having antimicrobial activity. It is resistant to change in the environmental conditions. So fungal infection won't be there, bacterial infection won't be there. So the durability and sustainability of dye is getting increased in the environment. And the main approach in this case is that there is a kind of slow poisoning happening in the nature due to this approach.
Might be possible because we don't know that. Silver uh, actually not differentiate that is animal cell it is good and that is bacterial cell is not good. So we cannot predict that.
Basically, what I can say. So as you told these concepts, so there are engineered nanoparticles, as you mentioned, NPs. So as already you mentioned that people use for antimicrobial resistance and also to improve the quality of the paint, and there are also it comes with a lot of other benefits. So the main thing is, after I mean that paint it has actually specific life cycle. Okay, so once it is introduced in the paints, of course, it actually has some good, what I can say, good effects maybe. But after some time, there's a lot of concern about its life cycle when it comes to the life cycle assessment. So let's say because paints have their lives after certain time, let's say after 10 years or maybe 20 years, eventually they will fade off. Okay, or maybe they will
And
they will leak.
properly come into the nature.
Yeah. So they will leak. So there is a possibility leaking or leaching of that nanoparticles, and after that leaking of, even though the concentration is lower, but it will come into contact with wastewater eventually, or maybe let's say any kind of environment, you can say.
Rainwater more specifically.
Rainwater. So that is the huge concern, and there are some papers which actually showed that silver nanoparticles, basically not only silver nanoparticles, they actually do affect. They perform toxicity studies, including phytotoxicity and cytotoxicity. So
But it has been known for the long period now.
yeah. So what actually my point of view is, there should be some regulatory assessment.
I would like to know your opinion, that approach that has been happening right now, it is appropriate or it is not good for us. Having said that, we have a long history of knowing toxicity of dyes and nanoparticles.
In my opinion, it is appropriate, but there should be some regulatory assessment for that. For example, if a company manufactures those ENPs, and if they are introducing into paints eventually, so that company should actually do some regulatory assessments.
What type of regulatory assessment?
They have to see how much concentration is actually. For example, if they must be having some amount, actually how.
Silver nanoparticles are not new for us.
Yeah, I know. So
If it was wanted to create, it could have been created.
yeah, that is true.
Your approach is very nice, but considering it is not a new category of contaminant and not a new category of emerging contaminant, it's been with us for a long period.
Strictly regulated.
Yeah, it is kind of way answer.
So that's what I said. So there should be some strict regulatory assessments, that they should do some life cycle assessment of those paints. They have to see about like how much that, eventually that is part of R&D. So how much of those quantities of, let's say, some part per million or some amount is actually leaking from the paints. So based on that, they have to calculate, and then they have to make some regulatory frameworks based on that, and then they have to introduce into paint. So I think that might be the solution.
Leaching of nanoparticles, it's been well studied.
Yeah, that is true.
It's not a new for us.
Yeah.
Okay, let's move on to the synthesis of magnetic nanoparticles.
Okay, first of all, we need to understand what is a magnetism and how it arises. Actually, basically, uh, magnetism arises due to the unpaired electron present in the matter. So spin, electron spin is very important. If the spin is aligned to the external magnetic field, it shows magnetism. If not aligned, so there is no magnetism present in the that matter.
In normal atomic configuration, spins are generally clockwise and anti-clockwise.
Clockwise and anti-clockwise, plus one, half plus and minus half. So that is not case. Depending on the nature of that particular matter, there are several types: ferromagnetic, diamagnetic, paramagnetic, and ferrimagnetic. In case of paramagnetic, it is a weakly attract. So some electrons align with the external magnetic field, and some are not. So there is a weak magnetism. In case of diamagnetic, they actually there is a no attraction at all against the external magnetic field. In case that we can say there is a one example known as a water molecule act as a diamagnetic, or molecule oxygen act as a paramagnetic. In case of ferromagnetic, it is highly attracted towards the external magnetic field, like iron, cobalt, and nickel, three elements shows ferromagnetic nature, extremely attracted towards the external magnetic field, because it is due to the that unpaired electron, or we can say it is domain which is perfectly aligned towards the external magnetic field. So there is a formation of ferromagnetism arise in the element of ferrous, cobalt, and nickel.
Can you?
In case of there is another case known as a ferrimagnetic. Here, interesting case. So some domain aligned with external magnetic field, and some are not. But alignmented domain is much more higher in number than that of the opposing domain. So there is a attraction towards the magnetism, external magnetic field. So we can say it is a magnetic, but not much as a ferromagnetic. The Fe3O4, that is a magnetite, fall into ferrimagnetic matter.
Can you explain how did you synthesize magnetic nanoparticles? For example, which solution did you use to make?
In my case, I actually particularly used a co-precipitation method, whereas I use Fe plus plus and Fe plus plus plus. There is a two state of iron. They are formed together, and there is a formation of hydroxide, and then it form after oxidation. There is a formation of ferrous ferric oxide, a stage where there is a both um, um, entity plus Fe plus plus and Fe plus 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 are in the same category, same area. So we can say it is a ferrous ferric oxide. That is a Fe3O4, a black color and highly ferrimagnetic material.
You mentioned about the spin of that electronic configuration. What is that?
Actually, spin of electrons, movement of electrons. So it may be clockwise or anti-clockwise.
Similar way, same pattern. Two atoms having same clockwise position and anti-clockwise position, or one is having clockwise or one is having anti-clockwise.
In that case, one is clockwise and another is anti-clockwise. There is a no attraction towards the magnetism.
But there is generation of energy.
Both spin aligned perfectly to the external magnetic field. So if there is a clockwise, both electrons should be in clockwise position. If another case, both electrons are in the opposite or is anti-clockwise alignment. So
What happens in case of diamagnetic, paramagnetic, and ferrimagnetic materials? So what is the spinning nature of electronic configuration?
in case of paramagnetic, some domains. We can say it is a spin electron towards the alignment of external magnetic field, and some are not. So they cancel each other, and there is a formation of small interaction towards the magnet. In case of paramagnetic, in case of diamagnetic, there is no any interaction. They not the electron not intact, or there is no any free electrons, and actually there is no any interaction towards the external magnetic field. In case of diamagnetic, water molecule is best example of diamagnetism. In case of ferromagnetism, they are highly aligned. The electron spin are electrons are spin highly aligned towards the external magnetic field. So there is a strong magnetism appears. In case of ferrimagnetism, some are not, or some are aligned, and some are not aligned. But the aligned uh, uh, aligned entity is more than that of the not aligned. So ultimately, it is strongly attracted towards the electromagnetic field, but not much as ferromagnetic property elements.
Okay. Tell us about your experimental conditions. How you synthesize nanoparticles? Which type of nanoparticles you synthesize?
Fe3O4.
How was the reaction?
It is a simple reaction. I use Fe plus plus precursor, that is FeSO4, and I don't want to tell you exact precursor name of the precursor,
That's
but
fine.
Fe plus plus and Fe plus plus triple plus. So both in a different proportion dissolved into water, and then addition of NaOH, which produces hydroxide ions, particular hydroxides, and then that hydroxide is then with the help of hydrothermal or whatever, we can heat that hydroxide into the uh, it become a oxides. That oxide hydroxide become oxides when we heat that into the furnace. It become a oxides of that corresponding Fe plus and Fe plus plus and Fe triple plus. So there is a formation of magnetite. In my case, there is a black color powder, and highly magnetic towards the external magnetic field, but it is very unstable. It get oxidized and form Fe2O3. It's a reddish brown color, and less magnetic as as much as it is a less magnetic than the Fe3O4. Fe3O4.
How did you check
And
the
you
man?
synthesize nanoparticles for Fe2O3.
I synthesize Fe3O4.
You synthesize
Fe3O4.
Fe3O4, that is magnetite.
Magnetite. And Fe2O3, it is a red color and less magnetic than that of the
Magnetite.
magnetite.
They are also magnetite.
Actually, I'm telling about magnetite, which is unstable. If we placed into the environment, it become a oxidized and to form Fe3O4, Fe2O3. That is a red color oxide.
But
So
earlier you said Fe2O3 is red colored.
Fe3O4, it is a black in color, and Fe2O3 is a red in color, which get when it is formed after the oxidation of magnetite. It is which is a black in color. In my case, I synthesize Fe3O4. That is a magnetite. It is a black in color.
How did you check the magnetism of your sample?
I think the name of the instrument is, I don't know, VSM, maybe VSM, with the help of VSM.
And which technique did you further use for more calibration?
Then XRD and X-ray crystallography, and TEM for the what is the morphological analysis. After that, I used that nanoparticle for the enzyme immobilization with the help of chitosan.
Which enzyme did you use? Chitinase? Chitinase?
Mm, Chitosan is it is a kind of polymer which like a duct tape. So after synthesis of magnetite, um, I use the chitosan as a polymer which act as a duct tape. So it stick on the surface of the nanoparticle, and after that I use a enzyme, that is a invertase enzyme,
Okay.
which stuck on the surface of the chitosan, which is coated to the nanoparticle. So there is a layer. There is a layer of chitosan. In one place there is a nanoparticle. On the outer side there is a enzyme. So we can repeatedly use enzyme many time, because it is very we can separate out that and magnetic nanoparticle from the mother liquor. So we can use repeatedly. That was my first paper, I think 2015.
So what are the results of your enzyme activity?
I found 5 to 6 cycle repeatedly.
Reusability.
Yes, reusability.
And can you tell me how, I mean, the mechanism, how, how did magnetic nanoparticles help to improve the catalytic activity of your enzyme?
Actually, it not help. It just like a we can separate out enzyme very easily from the mother liquor.
When you checked your enzyme activity, did you also use free enzyme to compare with?
Yes.
So what are the differences between the enzyme activity of your immobilized enzyme and free enzyme?
Somewhat it is a similar to free enzyme, but
Why it is similar? It should improve, no?
how?
I mean,
It is not actually that.
so what was the purpose of your immobilization?
Paper is to use repeated use of enzyme. That is the case.
So
After some time, the enzyme become uh, uh, released from the that chitosan polymer, because it is a non there's a no any kind of bond form between the chitosan and enzyme. It is a just non there is just hydrogen bonding. If we change the pH, it also releases from the that part that
Solution, the elution
But
will take
enzyme is
place.
quite complex structure. It can
Yeah.
interact with different bonds with.
yes, but
His application, I guess, for immobilization of the enzyme.
Yeah,
So it
that's
will
fine. But
contain.
immobilization again involves different types of
if
bonds.
there is a chemical bond, if there is a chemical bond, actually enzyme is very delicate structure. If there is any kind of chemical bond happen, maybe it loses their activity.
Yes, their activity is based upon the structural component.
It should be confined, but not chemical bonded to the
which method did you use for your enzyme immobilization?
after enzyme after the chitosan layer formation on the nanoparticle. I mixed that enzyme with that chitosan coated nanoparticle, and just we put that for overnight.
So, like for example, there are different methods of enzyme, like encapsulation, let's say, entrapment, okay, adsorption. So, which or cross-linking you can say. So, which
No, there is a no cross linking. There is a entrapment.
so
It is just stuck. Actually, chitosan act as a duct tape. It stick both the nanoparticle as well as enzyme.
but it must have formed some bonds, right? No. How did you encapsulate it inside? It must have found some bonds, right? Which bond did it form? Let's say some hydrogen bonds or some van der Waals
We can say it is a hydrogen bonding. Hydrogen bonding.
covalent bond.
No covalent bond. We actually, in my case, if there is a covalent bond form, maybe it loses their activity, because enzyme is three-dimensional structure. So it shows. So if there is a covalent bond form, some maybe there will be a deterioration there's 3D structure, and it
I don't
lose
think
their
so. Does it? I don't think so.
External, generally the motif region is more important in the case of enzyme. If it is shielded
there is
well,
a chance, there is a probability to lose activity. There is a probability.
when you talk about immobilization, successful immobilization, it is mean that there is a proper binding of enzyme to that particular structure.
No,
Yes, it's like that way.
there are lots of strategy, confinement.
Yeah, I just explained you.
There is a bond formation with the help of glutarate. There is a cross linker. We can use a glutarate which bind surface of the one part and it connect to the enzyme. But in there is another case, the encapsulation.
You use it encapsulation method.
Encapsulation where actually chitosan act as a polymer. Okay, it is a mesh like a polymer. So it entrap that enzyme entrap into the chitosan polymer.
Okay. I mean, I really can't imagine actually how did the system form. But
See, again, the reaction is happening between the chitosan polymer is there. Again, it is having a porous structure. So enzyme is embedded inside the structure. So water is there, enzyme is there, and chitosan is there. Chitosan porous structure, enzyme is embedded inside. Again, if you talk about bonding pattern, enzyme can have a different types of bonding pattern because it has a scaffold and it has a main motif region. If motif region is shielded well, it can interact the scaffolding material with the porous nature. And when we talk about polymers, polymers have different bonds, different functional groups. So they can interact with the enzyme in a very well manner.
Yes, it happens.
If you are saying that just putting a water into the solution again will take out the enzyme from the whole reaction mixture, it is not possible.
Whole
Reaction mixture. As you mentioned that there is involvement of hydrogen bond. When you talk about hydrogen bond, see, hydrogen bond is not the weakest bond. When it comes, it is a cumulative force. Okay, one hydrogen bond when interacts with one singular entity, it is weak. But when it comes with cumulative force, if you're saying water is there, number of hydrogen bonds are there.
yes.
If you see cumulative force, you need to add something that will hinder the cumulative force.
that's true. So again, like as you mentioned about, I mean, the enzyme activity did not increase; just it helped to shield basically or hold the enzyme, right? As you said, but it must have improved the enzyme activity, no?
Yeah, embedding with a proper structure, it should increase the enzymatic activity.
Why?
See, when you talk about aqueous solution, enzymes are not having a definite structure. You don't know, you don't observe the enzyme. It is distributed throughout the solution. It is kind of a dissolved state. But again, it is having a protein structure. So solidness is there. But still you cannot visualize where is the scaffolding matter, where it is the actual active site. If you embed enzyme in a system where active sites are exposed well, it can have a high efficiency other than the aqueous solution.
So bottleneck.
Actually, best example is Prasanna study. He has worked on the nanogel where he's had got good activity in the nanogel embedded with enzyme.
So the main thing is your active side must be exposed. So I mean, no, I don't know. Maybe in his case, active side must not be exposed. I don't know. Maybe or not confined well. It may be like that way.
Okay, let's end this. Thank you so much everyone for coming here and having this share time with me and giving a valuable suggestions and the knowledge that you have put onto this segment. I really appreciate it and I truly admire your knowledge and the perception that you have put here.
And we also thank you very much.
Yeah, thanks for hanging with us.
It really helped us even refresh our ideas and our understanding as well, and it variety of areas and variety of subject we have discussed. It was really nice to see and
To being here.
Yeah.
And
It's nice to.
it is fresh my knowledge and also
It's nice to have you guys on this panel, and it's my privilege to share this podium with you guys. And it's always. Thank you so much for this opportunity, Shubham. You say something, sir. You are the most senior here.
Thank you very much.